All right. It's a crazy, crazy day out there. We're going to go over DNA replication. Okay, so just to get your bearings, this is the S phase of the cell cycle. And so in the cell cycle, you remember you have your, and if we were in class, I'd be calling on people, you have your G1, you have your S, you have your G2, and then you have your M followed by cytokinesis. Okay, so right now today we're highlighting the S phase. And I'm going to try to move very quickly through this because it's a long process. And so if you have to pause the video, just pause the video. You need to draw this. You need to write stuff down. You need to be like really, really careful when you're working on this that you are working for understanding. So I'm trying to get everything set up here right for you so we don't have any weirdness. Okay, so in the S phase, so this is DNA replication, and it's making a copy of itself. And so the S phase, actually, S itself stands for DNA synthesis, okay? I'm going to write S phase here. And if you want to go ahead and write your notes that S is for synthesis, you can. That's completely up to you. Okay, so we have to learn some enzymes. So I'm going to draw a picture of what is happening. And then I am going to make a little list of the different enzymes. Remember, enzymes are proteins. And so you're going to be having 4 different proteins that help DNA replicate, make a complete, exact, identical copy of itself. This is important because each of the new cells, when the cell cycle is done, each of the new 2 cells need to have identical DNA in them. Otherwise, every cell in your body wouldn't have identical DNA, and that would be bad. Okay, so we're going to start off I'm going to draw like DNA open, like it's already been unzipped or unwound, if you will. Okay, and so then we're going to pull up here and I'm going to kind of helix it. So this is actually your DNA, right? So this is your DNA. And it's got your little nucleotides here. So if I wanted to write them out like A, T, C, and then this one would be T pairs with A pairs with T, G. So those are like all little nucleotides. And that's what those little dashes stand for. Okay, like those are all little base pairs. And here we have it like opened up. So we call this the replication fork. This is where replication happens. And so it's an opening, and it can go either direction. So it can go to the left, it can go to the right, doesn't matter. And let's give it some bases. Just pick some. You should be drawing this in your notes because if you were in class, that's what you would be doing. So you need to be doing it now. Okay, I think I got them all right. Sometimes when I do it quickly, I get how to pair bases. That's Chargaff's rule. A pairs with T, T pairs with A, G pairs with C, C pairs with G, blah, blah, blah. Okay, so the first enzyme we're going to talk about. So this is, remember, in the nucleus, in the S phase of the cell cycle, this is what is happening. Okay, it's so important that this is right, or else the cell will be messed up and that'll be bad. So I like to draw my enzymes as little shapes and things. So they don't really have this shape. They're like a blob. But I'm going to give this one the shape of an arrow because that helps me realize that it's actually moving in that direction. Excuse me. So this is helicase. Notice they all end with ACE because they are enzymes. So helicase. So what helicase does is it unwinds the DNA, and it does that by breaking hydrogen bonds, okay? So breaks the hydrogen bonds, allows the DNA to open so that the other enzymes can come in and make a copy of it, okay? If it's all wound up, it can't have a copy made of it. So helicase is the first one on the scene, and it makes that happen. Okay, we're just going to replicate one side first. Don't do both of them. They are replicated differently. So I have to show you different. We're going to do, it doesn't matter, top or bottom, doesn't matter. I always start with the bottom. So, but other videos I've seen always start top. So, but it doesn't matter. So what happens next is we are going to use a molecule, or I shouldn't call it a molecule, a protein called DNA polymerase. You may remember from transcription that there was one called RNA polymerase. But And so what it made was a polymer. That's what that polymerase. Polymer, meaning a long strand of repeating units, made a polymer of DNA, or the RNA polymerase made a polymer of RNA, but this is DNA polymerase. So it's an enzyme, ACE, making a polymer of DNA. We're going to use it, but the problem is DNA polymerase has some limitations. And one of its limitations is it just can't start like with this. Like with this DNA here, it can't just be like, oh, I know where to start. So it needs a primer. And if you've ever like painted a wall or a canvas or something, you have to sometimes prime it first. You have to get it like a starting coat, somewhere to start. And so the primer that comes in, and I'm going to draw like a little box or a little thingy, pretend like it's protein. I don't know what shape it is actually RNA primer. So A pairs with T. Oh, so it's U's. Oh, that is so strange because RNA really shouldn't have anything to do with this. This is just DNA making a copy of itself. But RNA you'll find in biology a lot of times is our little helper. So RNA primase, and I'm writing in all caps, but you know, they're not all caps. It's fine if you lowercase them. It creates a primer that helps DNA polymerase begin. Running out of space. Okay, that's what it does. Okay, so it's that little green thing there. Okay, so now we have two so far. So we have helicase that unwinds, and we have RNA primase that lays a little, and we call that a primer. So this little AUG here, AUU, that would be a primer. Okay, that's a primer. All right. Give you a second. Got all that? Oh, you can pause the video. I don't need to give you a second. Okay, so then we have next up, we are going to have, well, I wish I had another color. I don't know my colors at home. We're going to have now DNA polymerase. So DNA polymerase actually lays down pieces together, creates, I'm going to say lays down DNA nucleotides. Oh, I ran out of space. Using base pairing rules, Chargaff's rules. I'm going to write something that's going to make no sense to you. In a 5' to 3' direction. And then I'm going to explain it to you. Okay. All right. So C pairs with G. G pairs with C. A pairs with T. C, T, A, blah, blah, blah. So you're just going on and on and on. Okay. And this is our little, I'm going to draw a blob. Here's our little polymerase that's doing that. Okay. So what it does is it just lays down bases, and it's just like, hey, primase, cool. Thanks for the little primer. I got this, and it just goes zip and lays down bases, and it would just keep going. Follows helicase, okay? So it follows the same direction as helicase. And this one, okay, now I have a little issue because I said something here that is brand spanking new. So what the heck is 5' to 3' direction? Maybe you remember, hopefully you remember, back in the day when I would draw nucleotides, and they would look like this, like that maybe, yeah? And then I'd say, oh, and then another one attaches here. Okay. And it has another little base. And then, oh, these are upside down. Remember that? Anti-parallel. Oh, gosh. Anti-parallel. Doop, 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 doop. And then here's another one. Okay. I'm going to focus on this part for a second. All right. You look at my floor and my cords. That's nice. And then here's another one here. Okay, so these are, remember, this is the carbon. Still drawing, sorry, can't draw and talk. Oh, I forgot my phosphate. So this is a 5-carbon sugar, these little houses, okay? So remember, this is the sugar. Sugar, 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 sugar. Phosphate is the little dot. Phosphate, phosphate, phosphate. And then these are your nitrogenous bases, right? And then there's hydrogen bonds in between them, and that's what the helicase breaks. The hydrogen bonds crack. That's what helicase does. So if you were like thinking about like where this is, it's up in, this is so professional. Like this little chunk here, if I were to turn it sideways and put it, it would be like right there. Like there it is. There's the nucleotides. Okay. So 5' to 3', that's what we're dealing with now. Each of these five carbon sugars, so this deoxyribose here, you can number the carbons. All right, so I'm gonna do it with a different color. Okay, so this is actually oxygen at the top. I think it's fun because it kind of makes me think of like 0, because then I go clockwise and at every bend in the molecule, it's a carbon. First carbon, second carbon, third carbon, fourth carbon, and then the fifth carbon is up here attached to the phosphate. So that's a 5. Okay, so first, second, third, fourth, fifth. Why is that important? Because that is what, I just threw my marker on the floor, because that is what 5' to 3' is talking about. Okay, this is what they refer to as prime. Okay, so if I am a helicase and I'm coming along here and I'm going to unzip this, now notice I'm going to have to move like in that direction, or I'm going to have to come down this way, but I can't go this way on this side and that way on this side. I'm just unzipping this molecule, right? And so if I'm moving this way, okay, then I am moving from a 3'. Oh, sorry, you can't see my fingers in the way. 3' to 5' direction. So see that? Going from 3 to 5 this way. So look, these are upside down. So there's 0, 1, 2, 3, 4, 5 up there by the phosphate. And that's not 0, that's oxygen, but whatever. 1, 2, 3, 4, 5 up by the phosphate. So on this side, if I'm going from the bottom of my drawing to the top, then on the left over here, it is a 3' to 5' direction. That's the way this is going. On the right, if I'm moving from the bottom to the top, I'm coming to the 5 carbon first and then to the 3. So this is a 5' to 3' direction. Okay, so that's what I mean by this direction. So polymerase, DNA polymerase can only place nucleotides in a 5' to 3'. They can only do it that way. That is a limitation of their protein. Remember how proteins are a folded thing and they have like very specific shape? So this particular shape can only, put the nucleotides down if the nucleotide is facing a 5' to 3' way. It can't do it the other way, which makes it very difficult to like lay down one side or the other, because one side, remember, DNA is anti-parallel. Okay, so if this has been laid down up here, let's get back to here. So if this up here has been laid down in a really fast, easy direction, that means that these were laid down in a 5' to 3' way. So that means that this had to be the 5' side right here. This side did. And then this over here is the 3' side. Okay, oh, you cannot see that at all. So this is the 3' side, because my DNA polymerase could just lay those down really fast. So they're laying them down 5' 3, 5 3, 5 3, 5 3, 5 3, 5. I got this. Okay. And then this side, then this template here, this template that it used to make that copy was 3 to 5, because it had to be anti-parallel. So if this one's 5 to 3, this one has to be 3 to 5. Okay. That's a lot. That is a lot of information, but we're not done. So you can see, hopefully you can see right away, if this one was 3 to 5, then it's originally was bonded to that one. Remember, we opened it up. And so this one then is 5 to 3. That one's 5 to 3 because it had to be anti-parallel to this one down here. Okay, cool. I mean, not really cool, because now what happens? My DNA polymerase, it can't just lay bases down this way. It can't lay them down in a 3 to 5 direction. That's not it. That's outside of the scope of its abilities. And so what the primer has to do now is do these little primers, like going this direction. So it's like G, C, and then it's like, okay, I go this way. A, T, and it's moving this direction. And then it bounces back over here and it's like, okay, U, because it's RNA primase, A, and then now it goes this direction, G, T, and like that. And it makes these tiny little fragments. but it has to go this way and then bounce back, go this way and bounce back, and then go that way and then until it gets all the way to the end. It's really slow. We call that the lagging strand. Okay, because it takes so long, it lags. We call this one the leading strand because it's really fast. Okay, and we call those little fragments that are made right there, those little weird fragments, we call those, oh my goodness, I am so sorry for my non-professional video today. We call those Okazaki fragments, named after the researcher that discovered it. Okay, Okazaki fragments. They're these short little fragments that are made on the lagging strand because it can't just lay it down in one big strand. It has to be primed every single time. Okay, then your primer gets removed. So primers get removed. Let's get rid of the primers. And in their place, a different polymerase comes in and fills in the gaps with actual DNA. You have to have DNA. You can't end up with primer with RNA in your DNA. That's a mess. Okay, and then our final guy, our final enzyme is going to be ligase, which I like to think is like ligament. Okay, and that basically attaches the fragments together. Okay, so it attaches the fragments together. Okay, so ligase comes in and it's like, oh, you were once apart, so we're gonna just kind of glue all this together and make this one big happy family. Okay. Oh, I didn't label this one. So if this is 5' to 3, then the new one that's being made is actually going to run 3 to 5. Okay, and then you can see if this continues to go all the way down, you're going to end up with identical DNA. So even though this is a big mess, like, oof, what a mess. You can see that you have AT here and AT here and TA here and TA here and TA here and TA here and GC and GC. And so it's identical. When it finishes all the way through, all the way done, we call that sister chromatids. They are in the middle of the cell cycle. Okay, so while they're in the cell cycle, then let me show you. So when it's finished, you will have two identical strands of DNA. They look just like each other, and they are attached together with a centromere, and we call them sister chromatids. Okay, and so there's one and there's the other, and this whole thing is called a chromosome. Okay, why are they not two chromosomes? Well, we have a couple problems with that. One thing is they're completely identical, so they're not like different chromosomes. But the other thing is that your chromosome number as a human, you have 46 chromosomes. Okay, and that is part of our identity. Like, that defines us as a species. So if all of a sudden you're like, oh, just kidding, in the middle of the cell cycle, we actually have 92. It's like, well, then you're not even a human anymore. Like 46 chromosomes is what we have. And so never 96 or 92 or whatever. And so how we do this is we say, as long as they are in the cell cycle in an actively dividing cell, we call one chromosome two sister chromatids after it goes through the S phase. After it replicates DNA, then they are completely identical, should be, barring a few mutations, attached with a centromere, going through the cell cycle. Then we call those two sister chromatids and not separate chromosomes. Now, as soon as cytokinesis occurs, when the gel is split in half, then they become chromosomes in their own right. Okay, then they are chromosomes. Okay. All right, so these sister chromatids are created in the S phase of the cell cycle in a process called DNA replication, where we're just learning 4 enzymes, but there's like 8, but 4 enzymes help to create these, and you need to know all of those. All right, there's a review activity. I recommend doing it. It's not what I wanted to do. I wanted to model it in class, but it does help. Make sure that you know this stuff, that you can identify the enzymes, and that you understand how it all works, okay? I miss you guys. I hope to see you very soon. Take care of yourselves in this really sketchy weather. I'll see you soon, okay?